Hey, everybody. It's so exciting to be sitting with you today. I am particularly thrilled to be sitting with this group of people, with Michael Hammer, Miriam Meisler, Krishna Veeramah, JayEtta, and myself, Gabi, because this is a group who worked to make the discovery that SCN8A actually caused epilepsy in humans. And this month, March 2022, marks 10 years since the publication of their discovery in the American Journal of Human Genetics. So this is just an informal chat for the SCN8A community to get a little view into reminiscing with this group about kind of how this all came together and just reflecting back on the incredible progress that has happened over the last 10 years. So Michael, why don't you kick it off and tell us a little bit about what led you to start taking things into your own hands with Shay?
Yeah, sure. So, yeah, I'm excited to reminisce a bit about what was going on 10 years ago. Of course, 10 years ago is when the paper was published. And so there were some years preceding that in which this all got rolling. And it was motivated, at least my end of this. So you can hear each of us has our own history that brought us together. Miriam is the person who made the biggest dent in SCN8A in the world from a mouse and human perspective. So she was doing work from the 90s on, 1990, what did we decide was 1992. I didn't know anything about SCN8A in 1992. In fact, Shay was born in 1996, so I knew something about SCN8A before I knew anything about it, and that was I had a daughter with an undiagnosed form of epilepsy, and we worked with Dr. Talwar. He just did a great job. Given that we knew nothing, he just did his best to treat Shay with what he had in his experience, worked on other kids with similar symptoms, but not knowing what the genetic basis was. I, being a geneticist and had a whole career doing evolutionary genetics, and Krishna came to my lab in the 2000s at some point. Can't remember exactly the year, but we were interested in human origins and human diversity. But we did have a very active DNA sequencing program. And some of this is technology driven because what we ended up doing, jumping forward a bit, was sequence the genomes of our family. But so that had a whole technological background going, where the price of DNA sequencing actually became affordable around the time that we decided to do this. So that was another element that led to us doing what we did. But again, going back to the motivation, as great as Dr. Talwar is, Shay's epilepsy was hard to control. And especially when she turned about 4.5, she developed a new seizure type, which Dr. Talwar called late onset epileptic spasms. And they were very difficult to control. So while she was semi-controlled on valproate and lamotrigine, two commonly used drugs for epilepsies in children, all hell broke loose thereafter. And I just was very frustrated because I'm a scientist and I wanted to know the cause of her disease, and I wanted to know as a geneticist what the genetic basis was. And
what made you think it was genetic?
There was no signs of anything else, no environmental signs or injury. The birth was a bit difficult, but there was a cesarean, but there was no evidence. She had good APGAR scores, and there was no evidence that anything else had happened. So I don't know, maybe my bias is that genes play a role in a lot of these diseases, whether there's lots of genes playing a minor role or whether there's a single major gene. But I'm biased. I'm a bias, but Miriam, your question is a good one. I mean, I spoke with, before I did this, I mean, it took a while to get this organized. First of all, you're not allowed to study your own family, from a human subjects point of view. So it took months to get even the Human Subjects Committee to allow us to do this. I had to invite in a PI, a co-PI, who would consent my family. I can't consent my own kids. That's coercive. You know, they might get punished if they said no or something. So lose TV rights or something. But I also had sort of several meetings with colleagues, you know, our best and brightest at the university who are geneticists or who were very good at some disease, like Fernando Martinez was one I remember, who's an asthma guy who studies the genetics of asthma. And it's a complex disease. So, but you know, it was funny. I mean, I thought everyone would be very encouraging, but during these meetings, people were like, you're crazy. You're crazy to do this, you know. It was going to cost a lot of money. And what are the chances we're going to find anything? And I'll tell you, the difference between a scientist making a decision to go forward with a project based on the cost and the time and the likelihood of success. I mean, you've got to have better than a 50% chance of success generally before you design a project, right? Before you go forward with it. But as a father, I was doing anything that would help her by 1%, you know? So I had a very different perspective. I thought we'd tried so many things. We tried diet, we tried all kinds of vitamins and supplements and different drugs, you know, failed after failed. And then just trying sleep and lifestyle and every little thing that would maybe help her just 1 or 2%. And those were big gains. So my perspective was if we had a 1 or 2% chance of finding by sequencing the genome, that was going to be a worthwhile project. So anyway, cut to the chase here. I think the cost of whole genome sequencing in 2008 or 9 or 10 dropped from maybe 100 grand a genome to like 10 grand a genome or something on that order. It was dropping, but it was always way too expensive for me to do a family of 4 until I guess around 2010, 2010. And suddenly it dropped down to 10,000. And it wasn't like we were going to do it in our lab. We outsourced this to a company called Complete Genomics, which I don't know if they exist anymore. They had developed a novel way of sequencing the genome. And so once we got all of our ducks in a row at the university with the Human Subjects Committee, my co-principal investigator, and talking to Krishna and a few other people in the lab, like, how are we going to do this when we got the data? And this is where my memory gets a little bit vague, so Krishna can fill in here. But we had written when did we start writing our first computer code for pipelines to analyze genome data? Was that before we got in this project? Or was it, I don't remember.
No, we started, because it was a completely new type of data. None of us had ever used that kind of data before, especially the complete genomics. It's all sequence data, but it had a different format and all of that, right? So it's very different. So I was hired to do the primate stuff, right? And so we were getting what would now be called standard Illumina sequencing, but this complete genomics. So we had to develop something new. It's a completely different format. I think a lot of it stemmed from that Peter Vischer paper. There was a paper on 10 trios with, I think at the time it was called mental retardation, but now intellectual disability. And they found some success. And I think they've just done exomes, if I remember.
I don't think, wasn't it the paper by Lynn Geordie?
Well, there was a Lynn Geordie paper on a quartet that found a inherited disorder,
which of course also turned out to be his own family.
Yes.
Oh,
Yeah.
Nothing like motivation, is there?
Yeah, no, that was big inspiration for me, was that quartet paper, because we did a quartet. I think we might have been one of the first to follow in terms of doing a quartet, to diagnose any kind of disease whatsoever. I mean, much less a neurodevelopmental disease. But that didn't. I was just like, you know, you look back and you go, wow, we were very early on in using this technology. But that was just driven by a father's, you know, craziness to figure out what was going on to help his daughter. It wasn't driven by, positioning oneself as a scientist to be sort of on the cutting edge. We were just trying to find anything that would work. So I'm gonna stop talking for a while, but I'm gonna end my piece of this part of the story because we can pick it again up when we've discovered the gene and the call to Miriam that was so amazing that we'll never forget. In the process of, at some point we got started and I don't know if we had the sequences back from Complete Genomics yet, or we were in the process of analyzing code, writing code and putting this all together, and Shay suddenly passed away. And obviously that was devastating to me. So I disappeared from the scene for a while. I mean, I think I was down for the count until Krishna called me one day and said, I think we found the gene. And Krishna, I lived in a black cloud for several months there, and my memory is just completely clouded. So why don't you pick up, tell everyone what you were working on. So, so the primate thing, we were sequencing primate genomes, but what happened when I disappeared? And tell me, this will be the first time I've heard it.
Yeah, I think have to recall myself. So I was hired for this primate sequencing project, that you got the NIH grant for. I remember we were going to start working on things, and I think we'd had Robert Erickson had kind of got involved, right?
Yeah, he's the principal investigator, Robert Erickson, who I think Miriam might know.
An old friend of mine. Was he the guy you got to be the?
Yeah.
Oh.
Yes. He came from Michigan, I think.
Yes, he did. Yes.
Yeah.
I mean, I can't remember the exact order. I know I kind of organized some kind of journal clubs that were like 6 p.m. and wrangled people to go to them by giving them donuts and things. And we were kind of going through what was coming out in that area. I remember we read the Vischer paper. We started to think about how we would analyze it. And then I can't remember if that was before or after Shay died. I do remember when Shay died, basically, we just kind of waited, and we kind of waited for your kind of, should we keep going with this, or did you want to stop? And I remember you told us you wanted to keep going. And so we kind of, I think that's the point we really kicked into kind of drive, and we started really working on the code and things like that of how to process the data. Before, I think we may have had the data, but we were just kind of playing around with it and learning with it. And then, so we thought the first obvious thing was to look for de novo mutations because of that Vischer paper, and I wrote some code. I don't know where that code is. And Olga is the person that
Olga, yeah.
Yeah. So I got her to run it. And I remember she kind of was there late, and she goes, oh, I think there's something interesting here, because it turned out to be that SCN8A mutation. At the time, we were just trying to find a bunch of candidates. So Olga's the one that actually spotted it kind of first in the data from the pipeline that we wrote as something potentially interesting.
Olga?
Yeah.
Yeah, okay. Olga has passed away. She cancer. Yeah.
Oh, that's sad.
Yeah. Wow, I didn't know she spotted it first. Wow.
Yeah, because she ran the code, and we basically were just coming up with a candidate list of mutations and ones that were hitting our criteria, which ones were non-synonymous, which one were genes, which one had good coverage, and I'd wrote some basic filters to kind of weed those things out, because something we were always concerned about is false positives, right? That's because the sequencing technology, particularly then and even now, always made mistakes in the sequencing, and we were getting a handle on the fact that how do we parse out what's real and what's not. And this particular site had all the hallmarks of something that was real, and then it was in a gene that, at least to us naively, made some sense. And then I think I contacted you, Michael,
Yeah, no,
I
I remember that. I remember, I think we found the gene. That got me off my bench in my closet. And then we were like, okay, well, we'd done a lot of Sanger sequencing to rule out all the false positives. And I think there was something like 37 possible de novos, and it came down to one that validated.
Yeah.
Something like that. So one turned out to be real. So we were like, okay. And then we looked up SCN8A in the literature, and we found Miriam. But we also found that Trudeau paper that Miriam was on, the only known case of an SCN8A variant in human, didn't lead to epilepsy. So in a way, that gave us a steeper uphill battle to prove our case. But in any case, that was the time we called Miriam. And I don't know, Miriam, you want to remember?
Well, I was sitting right here at the desk where I'm sitting now. And I picked up that telephone, and Michael said, I don't know if you'd be interested in this, but you didn't tell me that it was your daughter until about an hour later.
Yeah.
But you said, we found this mutation in SCN8A. And I said, I've been sitting here for 20 years waiting for this telephone call, which was true. Because we had been searching for the human correlates of the disease. We had found the mouse gene when there was no sequence databases. And we were going by a linkage, and we were marching through this genomic sequence and sequencing introns and looking for coding sequence. I was in London when Dan Burgess called me. He was a graduate student, and he said, it's a sodium channel. We didn't know what was wrong with this mouse. We suspect, it had a neurological phenotype. So we found it was a sodium channel. And then we started screening human patients who had similar phenotypes to the mouse, which was loss of function. See, that was the thing. The mouse was a loss of function. The mice couldn't walk. And so we screened, they had tremor. We screened essential tremor families. We screened ataxia families. And that's how we got to the Trudeau paper, because that was a family with ataxia. That's how that family got to the doctor. And so we found that null mutation, that was a heterozygous null haploinsufficiency. And we published that, but we kept screening. I hate to tell you how many, and screening in those days meant amplifying each exon and doing Sanger sequencing. So we looked at a lot of DNAs. So when you called, I said, yes, we are definitely interested. And we had, of course, we had the cDNA ready, and we knew how to introduce mutations, and we had the collaborators ready. So I contacted the Waxman lab, and I, enlisted them to do the physiology. But I remember I made a visit out to you. Did you bring me out, or was I out there for some reason? But I remember meeting Krishna, and we met in that kind of darkish seminar room, and everybody explaining what they knew about remember that, Michael?
Yeah.
It was very real. It was very intense.
Yeah. Yeah, so let's talk a little bit about why we enlisted our collaborators that we did, the Waxman lab, and what we had to show in order to make our case. I mean, finding a variant in a gene that, in a gene that, was a neurologically related gene for a neurological related phenotype, but that's a stretch. So we didn't have any epilepsy cases to say, but that's how people these days will say, we have high confidence that a gene variant is associated with a particular phenotype or a disease because there are other cases, but we had no other cases to go by. So we had to show it physiologically that it made sense. And you, like you said, you lined up all of the investigators, but we had to prove that it was autosomal dominant. We had to show that it made a change in the channel function that was consistent with epilepsy. And that's what we did, right? You want to talk a little bit about what you did and what the Waxman lab did?
There were a couple of challenges because it's a very unstable cDNA clone. So just putting Shay's mutation into the wild type cDNA to have something to test was a challenge right there. And Janelle O'Brien was the student who had mastered that. And then, what was I thinking?
You got it into a cell.
Waxman lab, right? We sent them the cDNA, and they did the physiology, and it was clearly a gain of functions. See, that was what was different from what we had seen before. So that's why it had never been noticed before. And they did the physiology, and then it was really logic about how the nervous system works and why this gene with a gain of function being localized at the axon initial segment and being responsible for the initiation of the action potential. Why, if you had a hyperactive form of that channel, it would give you extra neuronal excitation, it would give you extra action potentials, which we later showed when we put this mutation into the mouse. And that's exactly what the neurons had.
And that means, and to translate that, that means that it caused seizures, right? That's the excitability.
Too often.
Right.
Seizures are an extrapolation from that, but what you could actually measure was, well, when we got the mouse, when we got Shay's mutation into the mouse and the mouse had seizures, that was the real, you could say the real proof of the pudding, but that was 3 years later.
But Waxman also put it into a rat hippocampal cell line or something, or was it a slice of some sort? What was it that he was able to do?
They looked at it was transfected mouse hippocampal cells, I think, yes. But the beautiful thing they could show was that the channel was clearly abnormal. And it had persistent current beyond what had ever been seen before. It was enormous. And it was to a neurologist at that moment, it became clear that this was causal. But how did we convince the American Journal?
How did we convince them?
We didn't have independent cases, but we did have this very compelling functional data. Sorry about that.
Yeah, that's incredibly telling then, that the mouse was really mimicking what you saw in Shay, right? These really intensive seizures, right? And that were, it just, I mean, what was that like for you, Michael,
Well, that was 3 years later. That was really intense for me. It's hard to separate, Shay from the, the experiments we were doing, in the sense of, you watch a mouse have a seizure and it's so much like, what Shay experienced. It was hard to keep the two separate, and it always has been, but that's okay. That's part of what motivates me. So that's okay. The mouse model was an extremely good mimic for people. Probably much better than in most cases for mouse models, I think. Which is always really positive, because mouse models can fail many times in trying to translate what you learn in the mouse to the human. But the closer the model is in terms of its characteristics, the better chance you have to translate. So I think it's been really useful. I think the American Journal, we ended up publishing that paper very quickly. I remember it got some tough reviews, but I think they were all reasonable, and we answered, and it got published very quickly. So
it must have been compelling. It must have been compelling.
Yeah, I mean, that's exactly 10 years ago, March 2012. Here we sit in the middle of March 2022. then there was kind of a little bit of lull in the action. I mean, that was great, but then what? So Miriam, that helped you move your career forward.
We've had this grant that's been supporting SCN8A since 1995, since the same paper. So it fit right into our program. And that was a mouse genetics grant. And so making the mouse, now making knock-in mice was not that, oh my gosh, I had a marvelous collaborator, associate Julie Jones, knocking in that mutation into the mouse genome was quite a challenge at that point in time. And in fact, we're now using that allele that she made, because she did it with TALEN targeting. And she had to introduce a lot of synonymous changes to keep the mutant from being digested again. And now we're finding that it's a really good target for a CRISPR knockout.
Oh,
So
really?
we're actually going to move that model forward and see if knocking out the toxic allele would be therapeutic.
So that's knocking
As a
out
proof of principle.
so knocking out the mutated form of
Knocking out the
SCN8A.
mutated form, that allele of Shay's mutation is a good candidate for. We have an sgRNA that's specific. So
She's still
it
giving
goes
that.
on. It goes forward.
yeah. So as you moved your career forward, Krishna and I decided a slightly different angle, and it goes back to the exomes that he was talking about, the Vischer paper. We went to Dr. Talwar and we said, give us your 10 most difficult cases that you haven't diagnosed, and we'll do exome sequencing on the 10. And I think 6 or 7 of them all turned out to be a result that was very informative. Some of them were known cases, a couple of SCN1As cropped up, but there were some unknown cases again that we discovered. And now looking back 10 years later, all of those have been proven and published in the literature. Every once in a while, Krishna and I get emails from people saying, oh, I'm working on the such and such gene that you guys published. Can you tell us what you know? And we don't know much because we just had that one case, and we're not specialists in those particular genes, but it was a very productive, and that was one of the first, whole exome sequencing approaches to diagnose epilepsies, the genetic basis of epilepsy. It was even before the,
Absolutely.
what was the big project that,
Epi4K.
Epi4K, yeah, we published just before they did. So little guys working in mom and pop shops were doing it. But that was exciting work for us, and proof of concept that this could be done for other people suffering with unknown causes of their epilepsy. So that went on for, I think we published that in 2013. But somewhere around 2014, I'm guessing that one of these genetic testing companies, I'm not sure which one it was.
I think it was GeneDx added it. That was the one we got our results with first.
Yeah.
Yeah.
So we weren't going to discover other people with SCN8A until it was being tested broadly, and we had no control over that. So I just know that somewhere around 2014 or 15, and Miriam,
to the epilepsy panel.
And we started getting phone calls and emails from people saying, oh, I've just been diagnosed, or my child has. And that was a wake-up call.
Yeah, because I remember, Elliot was born in late 2012, and we first did the epilepsy panel in 2013, and SCN8A wasn't on it yet. And so then we had to do the whole exome sequencing, and that still took I think that was back in the days when it took like 6 months to get those tests done. And so then when we got the diagnosis, they handed us the one article in clinic from, with that talked about like clinical cases. There was not really a whole lot out there, but then we went Googling and we found Michael and we found Miriam, and we all came together in 2015. that's what, well, JayEtta and I started Wishes for Elliot in 2014. And then without the SCN8A diagnosis, thinking, okay, we're going to work in rare and undiagnosed disorders because there's too many of us out there. And then we got the diagnosis and we said, okay, so we were working at the time at Children's National with Adeline Vandiver, who was a phenomenal clinician, really working more in the myelin disorders. But, we were in the room getting the delivery of the diagnosis, and she's like, well, we have no idea. We don't know what's going to happen. There's only a few other cases in the world. We don't know if he's going to get better or worse, or if, anything. We don't know a prognosis. And so we said, okay, well, that's insane. We got to do something. So she suggested that we, work on advancing the research and bring clinicians together and researchers together. And within about 5 months, I think Miriam and Michael and, a big group of other folks, Bill Guyard from Children's and John Schreiber and JP Johnson from Xenon, and who else was there? We had your collaborator in Japan. We had this incredible group of, Jack Parent working on iPSC, on IPS lines, and
then of course,
Ingrid, of course.
Ingrid.
all of Miriam's followers and students who are then, we borrowed a lot from the SCN8A community who were ahead of us. So we got them interested in SCN8A. And so we got a good leverage off.
Yeah, that meeting was really catalytic. It really brought people together, got to know each other, and that made a big difference.
Yeah.
That was very powerful. Yeah.
Together, that accelerated and brought excellent people into the research, both electrophysiologists doing cellular iPSCs and mouse models and zebra models. And it really got things going on the research end. And then the clinicians, well, that's taken more time because clinicians need to get experience with the patients. But once the patients started coming, we just attracted a wonderful group of clinicians who were interested as well. And as Gabi said at the beginning, we've got this standard of care treatment guidelines project where we've got people around the world, all the best in SCN8A clinicians, putting their heads together, trying to get consensus on what kinds of treatment guidelines. Because the more we learned about the disease, Shay turned out to be one subcategory of the disease that I would say she was a moderate gain of function. We know that there's severe gain of functions and milder gain of functions, and there's loss of function, and there are different categories of loss of function, some with seizures, some without. And so the disease has split into not just a spectrum, but now we can do endophenotypes or subgroupings of patients and seeing if there's some precision medicine approaches that we can take to address each individually. And that's the future, but that's sort of where we're at in this consensus process, because there's no one thing, one drug fits all. Clinicians try all kinds of things, what they're comfortable with, just like Dr. Talwar did in his day. They do what their experience tells them. And so if you look at the patients as a group, they're on all the, I just call it a cocktail of different medications. We know that very much so because we started an online registry in 2014. When we started getting these phone calls, I realized that we need to study other people than Shay. We need to study all these people that are coming in. And so we've got 7 or 8 years now of data collection for the registry, which has been extremely valuable to see the spectrum, the sort of the natural history of the disease, and just keep the thing going in a positive direction.
Yeah, and of course that registry has been a huge part of propelling the knowledge base around SCN8A forward and giving us opportunities to be candidates for things like, pharmaceutical companies being interested in this space. And it's nearly unheard of that within 10 years of having discovered a gene is pathogenic, that you have 2 clinical trials that are in the works. We've got, yeah, pretty phenomenal. We've got the neurocrine trials that started late last year, and Praxis Precision Medicine will be likely starting their trials later this year. And, you know, I think that's in no small part due to the work of the registry and this community coming together. I mean, there has been a very robust scientific and clinician collaboration happening. You know, we started in 2015, but I think people have really started to come together as a community ever since then to, you know, share mouse models, share ideas, link up to say, oh, let's do some cross assay work. Let's, you know, so it's been hugely, I think, also due to, you know, the research that you did, Miriam, to build the foundation, you know, of our understanding of SCN8A, and then your collaborative spirit to work with, you know, all these other scientists, and frankly, to build the SCN8A pool of researchers. I think we have like what you're like we call you the godmother of SCN8A, you know, mouse researchers, right? They've all come through your lab or collaborated with you.
A couple of my postdocs have stuck with it, yes. Taken it to their own labs, 3 of them, actually, 3, Andrew and JC
Right.
and Jennifer.
Mm-hmm.
Both
What
of us
about,
that-
wasn't Andrew Eskai in your lab long ago?
Andrew,
Okay.
yeah.
Yeah.
Yeah, and of course the mouse, the Shea mouse, was involved in the justification for the clinical trial.
Tell us about that. How did that, what did
They
that
just
look like?
published a paper. Did you
Came
see
out in eLife this week.
eLife this week, and the title has to do with effective in a mouse model, and that's the N1768D mouse.
Yeah.
That we sent them, so they contacted us about the mouse model, and we provided it to them.
Was that Xenon that reached out first?
Yeah.
Yeah.
Yeah. Johnson, JP Johnson.
JP. Yeah.
That's a whole other interesting story, because he was trying to develop a drug to help with Dravet syndrome.
Right.
And they thought by knocking down SCN8A it would help. And that's a whole new area that you've done some work in as well, Miriam.
Yeah,
showing
that does help.
SCN8A is a major player in excitability. So even other forms of epilepsy can benefit by modulating expression of 8A.
Right, right. So when the right drug comes along, there'll be a huge market, actually.
Yeah. 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 So he was, we had a great talk with him. We should probably have invited him into this, but that might have been got too many avenues mixing together. But he had a whole long history of talking. It's very expensive to develop, you know, to come up with a new small molecule or any kind of drug that you can get FDA approved. It's huge. But he'd been working on it for years. And then when he heard about our stuff with SCN8A directly affecting epilepsy, that inspired him to keep going as well.
And they're still working, even though they, you know, they sold the SCN901, right, which is now, you know, the neurocrine clinical trials. We know they're still working behind the scenes to keep at it.
To get a better one, to get it better. Yeah.
Right, to keep, innovating and pushing and studying to see if there's something better. So
Really an interesting story of human motivation, isn't it?
Yeah.
It is.
Yeah.
It just blossomed, you know.
Yeah, I wonder if for each of you looking back, I mean, here you are scientists, you have different specialties, different tracks that you were on of the work that you were doing, and taking this retrospective to imagine that early time coming together and how we've got this community globally, we've got families all around the globe, clinicians all around the globe, and these trials. What is it like to have really been the seed that helped all these families have some notion of what was going on, rather than, you know, always living with, oh, you have undiagnosed epilepsy? it's just so phenomenal, the impact and consequences of that seed of instinct that you had, Michael, and bringing Krishna on and then connecting with Miriam. It's just, it was such serendipity. And
if I
so
ever stop
impactful.
to think about how it all happened versus what we don't know still,
Right.
I'm just so motivated by, it seems like we know so little, but yet that's why we're doing this. So we have to stop every once in a while and see how far we've come, because
Right.
it's discouraging if you just keep thinking about how little we know. But no, I mean, how do you feel about it, Miriam? And Krishna too. I mean, your career is, you've got this one paper from 2012 that's now getting cited quite a bit. So, do people ask, what's this Veeramah? Who's this Veeramah guy? Or how does it impact your life in any way, if it does?
Well, I mean, I work on obviously very, very different things now in my lab. It's historical ancient DNA, but every year I actually have to, when we have the incoming set of graduate students into the department, I have to give a 15-minute show and tell where I describe what we do in the lab, our history and things. And I always spend 5 minutes talking about the SCN8A project with all the other things that we're doing. And I always tell the students, if I work on these fun things, but not much of it's important. And if I never achieve anything else for the rest of my career, at least I did that. And that I always felt that was super important. So I always tell them that every year.
That's really
That's
beautiful to hear.
That's really beautiful. I think it's just amazing the way the international scientific system actually works. I mean, we knew something and we put a paper into the American Journal of Human Genetics, and then the whole world knows, and other people pick up on it, and the companies pick on it. I think it's a tremendous justification for NIH funding, for basic research, and for the publication system and the international motivation of scientists. I just think it's a beautiful story.
It is, and we're biased, but I think it's true.
It's incredibly powerful. And I think that you have a global community of families now that would agree, because we're all together, because you all took the time and the energy and the passion to make these discoveries and help us understand the link, right? And get us closer, not just to each other and supporting one another and helping each other learn. I mean, I think one of the other incredible things that has come of this is that, Michael totally shifted his career after that, right? I mean, you had this really prominent career as a human, geneticist studying, ancient, forms of life and humans. And then all of a sudden you're like, okay, well, never mind. I have a mission now. And Shay is, it's beautiful because Shay drives everything you do. And she really is an inspiration for the whole community. But, now you, aside from doing the registry, you have a mouse lab where you study, and are finding all these really novel things about SCN8A that nobody else thinks about or works on, because, you have this unique perspective. But then also taking that research and what we're learning from the registry and linking that with these family meetings that we have every week. We meet with families from around the world and talk with them about what SCN8A looks like for them, what it means to in their life, what drugs are working, what comorbidities are they seeing, how development is going. And it's just built this incredibly powerful, bi-directional, give and take, where we're learning from families and they're teaching us about what SCN8A is. And you're able to kind of give back and say, I mean, on lots of these calls, you have families reporting back and saying, well, last time Michael suggested, something that we're seeing for some kids is that this might be a better drug because based on this. And then families report back and say, it's helping, or you're right. I should.
First, we always make sure.
Yes, of course, to ask your doctor about it.
Yeah.
But it's just this incredibly powerful no other community has anything like it. You know, a father who is motivated, who's a geneticist and runs a registry and does the research and, you know, brings everything together to build this global community of families who are learning together. It's really incredible.
Yeah, we're in a unique situation where we hear so many stories of what people are experiencing, that we know more about the disease than anyone, you know, any one person. So, you know, we're not doctors, we're not MDs, but we have insights. And so now we're working with doctors and trying to transmit some of that, get some of that flowing among them. But yeah, it's you kind of just go where the energy is going. I mean, I had a lovely career, and I think it was a fine time to move on. It was hard to compete because of people like Krishna who, you know, the field moved in a whole different direction that was beyond my capabilities. So it was a good time to take a left turn. But you know, I took that left turn just because my Shay told me to. I mean, that's the way it's the easiest way to say it is I'm just following marching orders from her. So I'll keep doing it as long as it's helpful.
Well, from a family perspective, it's not only so seminal that you identified the gene, but the leadership that you've provided, bringing the community together and supporting these dialogues. It's so empowering for families that have so little they can control. Their lives are so challenging, and there's so much unknown about how to treat and improve the lives of their kids. But you create a sense of really being part of the solution by helping families really realize that their knowledge, their experience is the key to expanded understanding. And in a life that's so out of your control,
Yeah,
it's really wonderful to have families feeling like they're the citizen scientists,
yeah.
really bringing their unique knowledge to the expansion of understanding.
Well, that just kind of, maybe this is a good place to wrap it up. I mean, what Miriam said about the scientific enterprise, you know, the taxpayers pay for the NIH to fund us. And then in a sense, we're completing the circle by having the families being involved to the extent they are. And so that's the give and take. That's so nice to be part of.
Yeah, and I think one of the other really important things is the, you know, the alliance, the International SCN8A Alliance that, you know, Michael and JayEtta and I co-founded a little over a year ago, is that it's giving families these opportunities to engage with researchers and researchers to engage with families and pharma. You know, it's kind of having families at the center of everything and be able to contribute and engage, you know, in different ways. Like when we had, you know, Miriam, we're going to invite you to an 1872 meeting soon, but, you know, we had Andrew Eskai and Jen Wong join an 850 meeting, and it was so powerful. Researchers who studied this in a lab, you know, created a mouse model for that particular mutation, and then the families to be able to say, well, this is what we see. And Andrew and Jen were just so excited to get back into the lab and take what they saw from families, right? This is what the actual presentation is, and say, well, okay, how does that translate to the lab? And so I think there's this beautiful synergy now, you know, 30 years after you started studying SCN8A, Miriam, and 10 years since the discovery of the gene as causing epilepsy, that it's really an incredible point that we're at right now, an inflection point, right? It feels like, you know, we've got clinical trials, we've got all of this incredible work happening between families and pharmaceutical companies and researchers, and it just feels like we're on the path to something here. And hopefully soon, you know, being diagnosed with SCN8A means that there's answers, right? For a lot of us, when we got the SCN8A diagnosis, it meant no answers. So
the persistence and the collaboration that really got us here. So we are forever grateful, as is the whole community. And we thank you for your work.
Well, your work as well. Thank you all. Thank you all for coming together today. Krishna, I definitely want to talk to you more about what's happening in your life. So let's get together soon.
Definitely. A lot of change.
Always great seeing you, Miriam. So
Good to see everybody.
Yeah. Thank you all. Take care.
Thanks, everyone.
have a great
Okay.
weekend.
Be well.
Bye.
Bye.